Okay, this is the Professor, CBMJ PhD, and today's Thursday, October 26, 2023. It is 12:19 and 10 seconds a.m. Eastern Standard Time, and today's research, cancer metastasis. So let's hop to it here. Nanoparticle vaccine could curb cancer metastasis to lungs by targeting a protein. Date October 18, 2023. Source: University of California. Engineers have developed an experimental vaccine that could prevent the spread of metastatic cancers to the lungs. Its success lies in targeting a protein known to play a central role in cancer growth and spread rather than targeting the primary tumor itself. Metastasis is a process involving the migration of cancer cells from their primary site to other parts of the body. Recent studies have identified S189, a protein typically released by immune cells, as a key player in this process. Its normal role is to regulate inflammation. However, an excess of S189 can attract cancer cells like a magnet, causing them to form aggressive tumors and facilitating their spread to other organs, such as the lungs. The scientific investigators and microbiologists developed a vaccine candidate that can modulate the levels of S189 when it goes haywire. When injected subcutaneously, the vaccine stimulated the immune system in mice to produce antibodies against S189, effectively reducing the protein levels and minimizing cancer metastasis to the lungs. The vaccine also increased the expression of immune-stimulating proteins with anti-tumor properties while decreasing the levels of immune-suppressing proteins. The S189 is known to form what is called a pre-metastatic niche within the lungs, creating an immunosuppressive environment that allows for tumor seeding and growth. And by reducing the S189 levels, they can effectively counteract the formation of this pre-metastatic niche, leading to a reduced attraction and increased clearance of cancer cells to the lungs. And this is a clever new approach to vaccination that we are not targeting tumor cells, but rather the tumor microenvironment, so that it prevents the primary tumor from making new tumors. The vaccine consists of nanoparticles made from a bacterial virus called Q beta. The nanoparticles were grown from E. coli bacteria and isolated. Afterwards, a piece of the S189 protein was attached to the surface. How it works is that the Q beta virus nanoparticles act as a bait for the immune system. The virus is harmless to animals and humans, but immune cells recognize it as foreign and get fired up to attack the search for a pathogen. When the immune cells see that the virus nanoparticles display a piece of the S189 protein, they produce antibodies to go after that protein. An advantage of using antibodies is that they help keep the levels of the target protein in check. With this form of immunotherapy, they are not necessarily knocking out all of the protein, but they are reducing the levels everywhere. The vaccine was tested in metastatic mouse models of melanoma and triple-negative breast cancer, an aggressive and hard-to-treat cancer type. Healthy mice were first administered the vaccine, then challenged with either melanoma or triple-negative breast cancer cells through intravenous injection. The vaccinated mice exhibited a significant reduction in lung tumor growth compared to unvaccinated mice. In unvaccinated mice, the injected cancer cells circulated throughout the body and eventually homed in on the lungs to form metastatic tumors. The researchers know that this vaccine strategy combats tumor spread, not the primary tumor itself. While S189 does get overexpressed in certain primary tumors, it is mainly indicated in metastatic disease and progression. The protein is involved in the formation of immunosuppressive tumor microenvironments. Therefore, they found that their vaccine is much more effective at reducing metastasis and not in reducing the growth of the primary tumors. Another set of experiments demonstrated the vaccine's potential to offer protection against cancer metastasis after surgical removal of the primary tumor. The rates for triple-negative breast cancer tumors who received the vaccine post-surgery demonstrated an 80% survival rate, while 30% of unvaccinated mice survived after surgery. As these findings are the most clinically relevant as they closely model what could happen in real-life scenarios. For instance, a patient diagnosed with aggressive cancer who undergoes surgery to remove their tumor may be at risk of recurrence and metastasis to the lungs. The scientific researchers envisioned that this vaccine could be administered post-surgery to prevent such recurrence and outgrowth of metastatic disease. And before the vaccine can progress to human trials, more comprehensive safety studies are needed. S189 is an endogenous protein within the lungs, and there are a lot of data out there that demonstrate what happens when S189 is abolished. They know that S189 is important in the clearance of pathogens, and future studies should better test whether reducing S189 levels decreases the patient's ability to fight infections, especially in cancer patients who may have weakened immune systems. And future work will also explore the vaccine's effectiveness when combined with other cancer therapies, with the aim of improving the efficacy against hard-to-treat cancers. And this is a professor of microbiology, bioinformatics, pathophysiology, and teacher of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.